Hello everyone, my name is Arunabha. I'm a postdoc based at University of California, Riverside, working with Dr. Georgios Vidalakis. In this talk, I'll be presenting our former PhD student Shihua Tan's work on citrus Enation virus and investigations into its two distinct suppressors of RNA silencing. CVEV is a positive sense single-stranded RNA virus in the inamovirus genus of the Solemoviridae family. It encodes an approximately 6 kilobase pair genome, encoding 5 open reading frames, and produces isometric virus particles approximately of 28 nanometers in diameter. CVEV induces two main disease phenotypes in citrus: veination, which manifests as raised veins or papillae-like structures on the undersides of leaves, as shown here on the right in Mexican lime, and woody gall, characterized by knotty outgrowths on branches or trunks, as shown here on rough lemon. Although its economic impact on citrus is considered minor relative to more devastating pathogens, CVEV is widespread and can infect virtually all citrus varieties. It spreads primarily through grafting and aphid vectors in a persistent manner, making it important to understand precisely how the virus overcomes plant defenses. To give you a brief refresher, in plants, RNA silencing is a robust antiviral strategy. The plant recognizes viral RNA, processes it into small interfering RNAs or siRNAs, and then uses those siRNAs to target and degrade foreign viral genomes or transcripts. As a very effective response, viruses produce VSRs to interfere with different steps of this RNAi pathway. Some VSRs bind the siRNAs directly, others bind the host's dicer or Argonaute proteins, and still others direct these proteins for degradation. This schematic shows a simplified model of the plant RNAi pathway, and these are some well-studied viral VSR examples and the various stages of the RNA silencing pathway where they mediate host RNA suppression. In this talk, I will guide you through how two known CVEV encoded VSR proteins manipulate different facets of this antiviral host machinery. For this study, there were two main objectives. The first objective was to confirm the VSR activity of the two CVEV encoded VSR proteins, P0 encoded by ORF0 and coat protein encoded by the ORF3 in an experimental host. The second objective was to conduct experiments towards mechanistic characterization of these two CVEV VSR proteins. This included identifying potential host RNA or protein targets involved in their VSR activity, pinpointing regions of amino acid residues that may be critical for VSR functionality, and finally, investigating molecular interactions of these VSRs, which host proteins or RNA species are directly involved in these suppressor functions. For this work, we used CVEV isolate VE701 being maintained in a sweet orange tree at the Citrus Clonal Production Program at UCR. All experiments being described in this talk were conducted in Nicotiana benthamiana, both wild type and the GFP transgenic line 16C. The expression vectors used in this study included CVEVP0 and coat protein cloned into a 35S driven binary vector and transformed into Agrobacterium tumefaciens strain GV3101. Targeted ORF deletion mutant constructs were also generated, similarly to pinpoint essential motifs or domains in these VSRs. We also used a PVX-based expression vector, PGR106, to assess if the CVEV VSRs could boost PVX accumulation and disease symptoms in planta. All constructs which were built for this study were delivered into our experimental plants via standard agroinfiltrations. This entailed co-infiltrations with a 35S driven GFP construct for the VSR constructs and solo infiltrations for the PVX-based assays. For data collection, we used RT-qPCR assays targeted to in-planta GFP and PVX transcripts, normalized by the housekeeping gene NBPP2A from Nicotiana benthamiana. Northern blots with DIG-labeled riboprobes allowed us to visualize in-planta GFP, DS, and siRNA levels and PVX expression. To visualize RNA protein interactions in this study, we tagged P0 and coat protein with flag or CMyc for Western blots and immunoprecipitations. Investigations into potential RNA protein interactions were conducted in this study by RNA immunoprecipitation experiments as well as RNA protein pull-down assays. Now on to the results. This slide summarizes our first major finding. which you can see in these pictures of the infiltrated plants, where we confirmed that the CVEV-P0 suppresses host GFP RNA silencing both locally in the infiltrated leaves and systemically in the apical leaves. CVEV coat protein, on the other hand, acts mainly at a systemic level. We see bright GFP fluorescence in CVEV-P0 co-infiltrated areas at 5 dpi, while CVEV-CP fosters GFP signal in upper leaves by 14 dpi. The cucumber mosaic virus 2b and the citrus leaf blotch virus movement protein were added as positive VSR controls here. These visual observations were further validated by RT-qPCRs targeting GFP mRNA in both local and systemic Benthamiana tissue. Here, in case of the local infiltrated tissue, with CVEV-P0, you can see that the GFP mRNA levels are much higher than the CVEV coat protein. However, in the case of systemic tissue, both CVEV-P0 and coat protein infiltrated plants exhibited high levels of GFP mRNA in planta. This on the bottom left is a GFP siRNA northern blot that visually shows highly reduced siRNA levels in local tissue in case of infiltration with CVEV-P0, whereas there is abundant GFP siRNA present in local tissue in case of CVEV coat protein infiltration. Overall, these results confirmed the VSR activity of both CVEV proteins, with CVEV-P0 exhibiting local and systemic RNAi suppression, and CVEV coat protein or ORF3 exhibiting mainly systemic RNAi suppression. Now on to the results of the PVX-based infiltration assays. As a reminder, we inserted the CVEV-P0 and the coat protein into a PVX-based expression vector to see whether these proteins could increase in planta PVX infectivity. As shown in these photos here, Benthamiana leaves expressing either P0 or coat protein in a PVX vector boosted PVX symptoms in infiltrated plants, in parallel with the CLBV movement protein positive VSR control, as well as the PVX wild type. These plants showed stronger mosaic and leaf distortion symptoms. Simultaneously, RT-qPCR assays targeting PVX RNA revealed significantly higher PVX expression in both of these cases. The corresponding frameshift mutation constructs for both CVEV ORFs included as controls in this assay confirmed that it's the protein function rather than the mRNA sequence driving this effect. To pinpoint which amino acid residues may be essential for RNAi suppression by the CVEV VSRs, we created several targeted deletions within the P0 and the coat protein ORFs, starting with the P0 results first. The deletions are indicated with a delta symbol followed by a number range representing the deleted amino acid positions. Here, in case of CVEV-P0, it was seen that in agroinfiltration assays, the local GFP fluorescence via local RNAi silencing by the wild type P0 was curtailed in case of 3 mutants. These were D2, D6, and D7. This result was further validated by RT-qPCR assays targeting GFP mRNA in the infiltrated tissue. In this graph, you can see how the relative GFP abundance was lowest but not completely abolished in case of mutants D2, D6, and D7. These mutants respectively had deletions in amino acid residues 36 to 83 in D2, 176 to 210 in D6, and 211 to 245 in D7. In the systemic context, however, the agroinfiltration and RT-qPCR results both indicated that these mutations in the CVEV-P0 ORF did not significantly hinder systemic RNAi suppression activity of this protein, with visible GFP fluorescence in all cases of systemic leaves of infiltrated Benthamiana plants and significantly high GFP abundance detectable by RT-qPCR, respectively. Overall, these results showed that CVEV-P0's amino acid residues 36 to 83, corresponding to an F-box motif, which I'll describe later, and residues 176 to 245 are crucial for local silencing suppression, but not for systemic silencing suppression. Now, in case of the CVEV coat protein, we did a similar deletion analysis of the open reading frame. Agroinfiltration assays showed visually curbed GFP expression in systemic tissues for the deletion mutant D3. This visual observation was further verified by RT-qPCR, where you can see highly reduced GFP abundance for the D3 treatment, where amino acid residues 68 to 100 were deleted. Overall, this result shows that the CVEV coat protein amino acid residues 68 to 100 were critical for systemic RNAi suppression activity. Deleting that region highly reduced its RNAi suppression ability systemically. Now moving on to the mechanistic experimental results for these CVEV VSRs. On this slide, we see the co-immunoprecipitation results that highlight how the CVEV P0 protein and its mutants interact with Arabidopsis thaliana skip1 or AT-skip1. Skip1 is S-phase kinase associated protein 1 involved in tagging certain proteins for degradation. In this case, host RNAi associated factors. Now let me walk you through this figure. In the top half of this chunk, each vertical lane shows a different combination of flag tagged P0 or the Fbox deletion or the triple site mutant, plus the C-mic tagged AT-skip1 added to the blot lanes below. Their presence or absence is indicated by the plus or minus symbols. Now the input blot here is the protein input, where we confirm that the anti-flag antibody successfully detects the flag tagged P0 proteins and the anti-C-mic antibody detects the C-mic tagged AT-skip1 in the protein extract prepared from the infiltrated tissues. This red outlined bottom chunk is the main immunoprecipitation result. In this IP blot, the reaction mixture was probed with anti-flag antibody to precipitate flag-tagged proteins and any co-precipitates. We see in the lower lane that wild-type P0 and the triple site mutant were still able to bring down AT-skip1, whereas the delta 36 to 83 mutant fails to do so. The Fbox motif, which is located in this delta 36 to 80 region, is crucial for binding AT-skip1. And without that region, as in the delta 36 83 mutant, the interaction is lost. Now, via a second immunoprecipitation experiment, we probed into whether the CVEV P0 can directly interact with Argonaute 1 through its direct binding with AT-skip1 by testing the D6, D7 deletion mutants described previously. Just like in the last IP blot, the first half of the image here illustrates the different combinations of flag tagged P0 or deletion mutants added to the blot lanes below, their presence or absence being indicated by the plus or minus symbols. The middle chunk is the input blot, again confirming that the anti-flag antibody successfully detects the flag tagged P0 proteins and flag tagged NB Argonaute 1, which is the Nicotiana benthamiana Argonaute 1 in the protein extract prepared from infiltrated tissues. This red outlined bottom chunk is the main immunoprecipitation result. In this IP blot, the reaction mixture was probed with anti-flag antibody to precipitate flag-tagged proteins and any co-precipitates. We see in the lower lane that wild-type P0 and the amino acid 176 to 210 deletion mutant still bring down NB-AGO1, whereas the delta 211 to 245 mutant fails to do so. Together, these insights show how beyond simply recruiting AT-skip1, shown in the previous immunoprecipitation blot slide, CVEV P0 physically binds Argonaute 1 via amino acid residues 211 to 245. Overall, this two-step mechanism effectively disables a core component of the plant's RNA silencing defense. Finally, we tested for direct binding between the CVEV VSR proteins and DS or siRNAs using in vitro labeled GFP DS RNA and siRNAs and performing an RNA protein pull-down assay. The images shown here are of the final Western blots developed with the RNA protein pull-down products. The protein input blots show the relevant proteins present in the reaction. However, in the RNA protein pull-down blots, neither the CVEV P0 nor the CVEV coat protein was pulled down with GFP DS or siRNA, unlike our positive controls shown here, which is the turnip crinkle virus P38 and tomato bushy stunt virus P19 proteins. This result clarifies that P0's primary route is targeting host proteins like Argonaute 1 or SCIP1 rather than physically binding DS or siRNA molecules. However, CVEV coat protein also doesn't appear to bind either DS or siRNAs in its RNAi suppression mechanism. In conclusion, CVEV encodes two distinct VSRs, the P0 encoded by ORF0 and the coat protein encoded by ORF3. P0 suppresses both local and systemic silencing, whereas coat protein primarily acts systemically. Both CVEV-VSR proteins boosted PVX symptoms and titers in the PVX-based assays, confirming they promote in-plantar virulence. Deletion mutation studies demonstrated that CVEV-P0's FPOX domain, residues 36 to 83, and residues 176 to 245, are critical for local RNA suppression, whereas the coat protein uses amino acid 68 to 100 for systemic VSR activity. Mechanistically speaking, CVEV-P0 is capable of interacting with AT-skip1 and target Argonaute 1, thereby directing Argonaute 1 autophagy without interacting with any DS or siRNAs, whereas CVEV coat protein appears not to bind any RNA or host factors. Overall, understanding these suppression strategies informs us how CVEV persists in diverse citrus hosts and also suggests possible targets for breeding or biotechnological control. I thank all of the funding sources supporting this study. Please feel free to email me any questions at the address shown here, or you can direct them to my colleagues in the audience. And finally, I thank the IUCV organizers for this presentation opportunity. And with that, I conclude my talk. Thank you for listening.